Hello, my name is Anna Garrison, and I'm going to be sharing my research that I've conducted over the last couple years in the JOSTI lab. So the first thing I want to introduce is what a cycloparaphenylene is. What it is, as you can tell by the name, it's a cyclic structure strung together by para-linkages of phenyl groups. So what cycloparaphenylenes have is they have really cool photophysical properties, including a common absorption across all sizes of CPPs, and then a red-shifted emission. And what I mean by red-shifted emission is as you decrease the size of the hoop, you actually get a red-shifted emission or fluorescence. And another really cool CPP that you can make is a alkyne-containing CPP. So these alkyne-containing CPPs essentially just have an alkyne embedded into the CPP. And what's really cool about having this functional group is that it provides the means of reactivity of the molecule. And so we can actually change the structure of the CPP to actually modulate the reactivity by a couple of different ways. The first being the size. So if you decrease the size of the hoop, aka you just remove a couple of these phenyl groups from, say, maybe an 11 containing CPP to a 7 containing CPP, you're actually going to increase the reactivity because you're going to introduce a lot of strain on that alkyne. Another modulation you can do is an electronic modulation. So you can append these fluorine groups, which are super electronegative, and they're going to destabilize the alkyne and thus, increase the reactivity. You can also do a strain modulation. Instead of having all of the phenyl groups connected by a para linkage, you can introduce a meta linkage into the CPP, which essentially introduces like a kink into the molecule, which drives the strain right localized on the alkyne and increases reactivity. So you might wonder, what can I do with these alkyne-containing CPPs? They're super reactive, but what could I be using them for? And that is exactly what my project was. So I wanted to look at if CPPs could be used as biological fluorophores, and how well could they be used? And so what I did was I essentially hypothesized that via this, what we call a SPAC reaction, which is the strain promoted azide alkyne cycloaddition, that I could link an azide with the alkyne, a strained alkyne, and essentially make this linkage forming this triazole and essentially fluoresce the biomolecule because the CPPs are fluorescent. So how I sought this out was I first synthesized the 9+1 CPP. I won't go into too much detail about the synthesis, but essentially I started with a couple commercially available reagents. I used a lithiation addition in many different ways to make carbon-carbon bonds between the different rings. I also incorporated test groups to protect alcohol groups to kind of force the curvature of the molecule. And what I did was I created 2 coupling partners. And I then subjected my coupling partners via a Suzuki cross-coupling reaction, and I formed what we call a macrocycle, which is very similar to a CPP, but it still has these large bulky test groups, and it's not aromatic, obviously. And so what I did was I removed those test groups and aromatized the molecule to form the 9+1 CPP. So I explored the 9+1 CPP in my study because, with all these modulations you can do on the structure, like I showed, the 9+1 CPP is a nice balance between, something that's reactive, but also not unstable. And so I synthesized these for my experimentation as they're used for a biological fluorophore. And so the first experiments I ran were essentially stability experiments, which just tested the stability of the molecule in what we kind of consider like a cellular environment. So a cellular environment is full of moving parts. There's tons of different biological nucleophiles or things that it can react with. There's also things like pH and temperature you have to keep in mind. And so the first stability experiment I ran was just incubating this 9+1 CPP in deuterated DMSO just at physiological temperature. And I looked at the stability of the CPP just at physiological temperature. I used proton NMR spectroscopy to look at the structure of the CPP over the course of 7 days. And as you can tell, these peaks are very well defined pretty much after 7 days. And so that just showed that minimal degradation occurred. There's a little bit of there's a little bit of degradation here at the baseline, but overall, this was a very successful experiment. The other stability experiment I ran was incubating this 9+1 CPP with glutathione, which is essentially a biological nucleophile. It has lots of groups that the CPP could ideally react with that could be in a cellular environment. And then I monitored it over the course of 24 hours via, again, NMR spectroscopy. And as you can tell, the peaks became very, ill-defined. A lot of peaks formed here at the baseline. And what this showed was that a reaction had occurred after, looking into the literature and a little bit of analysis. We realized that the CPP actually reacted with the thiol group in the GSH molecule, which is actually another type of click reaction. So our experiment wasn't necessarily successful, but it did kind of introduce to us, different types of click reactions that these alkyne CPPs could be used for. But in terms of my exploration in a biological environment, I don't think that this result would hinder my results in general, just because of the cellular concentration of thiol groups in a living organism. So the next step was to actually, click a CPP to a biomolecule. And so what I did was I incubated, again, the 9+1 CPP with a azido sugar. So you can see the azide is right here, and there's a lot of acetyl groups. And here would be my predicted product. So after running this for 48 hours in a solution of toluene at room temperature, we analyzed the product via proton NMR spectroscopy. And what these peaks represent are the protons in the product. And after, analyzing this peak pattern, the splitting, we were able to determine that this proton NMR matched exactly to the predicted product. So this was a very good piece of evidence that we had indeed made our product. We also used mass spectrometry, which if you can see in these boxes right here, the calculated mass, so the predicted mass of our product, practically matched the mass of what we essentially made well. So this was also a very good indicator that we made the predicted product. So this was, the first account of clicking a strained-alkyne CPP to a biomolecule, which was very exciting. But as I said before, this experiment we ran in a test tube in 48 hours. And the next step would be to actually run these in living cells. And so that is what I did. And so for these experiments, we used a known metabolic pathway that was discovered by the Bertozzi group. And what happens is, you can treat cells with this certain azido sugar, and when they digest the cells, they install azides onto their cell surface. So if you can imagine a cell with azides on their cell surface, you can actually treat a cycloparaphenylene to the cell, and ideally it would click the azide to the CPP just on the cell surface and fluoresce the cell surface. And so that's what we sought out to do with our collaborators at OHSU. And we have here the results of the experiment. And if you can see, we see a lot of donut-looking fluorescence, which is a really good indicator that we did in fact fluoresce the cell surface of the molecule. And the panel to the right actually shows a positive control of another commercially available alkyne probe that we used. And this was very good in evidence that we did label the cell surface on a living molecule, and we're able to, perform these reactions with living cells without, being too cytotoxic. And yeah, so the future directions of this experiment and this project as a whole is just to repeat this cell labeling experiment with a nuclear stain and also some proper controls for publication. So we could essentially, treat these with a CPP that doesn't contain an alkyne, and we would hope to see no fluorescence. So that is more publication quality controls that we're looking for, as well as potentially repeating this experiment with different modulations of CPPs. So like I said, I used the 9+1 CPP because of its good balance between reactivity and stability, but ideally we could potentially make a CPP that's more reactive and maybe more fluorescent, has a higher quantum yield. So those are some ideas for the future. But with that being said, I want to thank the Josti group and Ramesh Josti, as well as a huge thank you to my mentor, Julia Fair, for being so encouraging and helping me with all of this research over the past couple years. So thank you.